We are going to conclude the workshop with the last presentation, which is given by Simone Ciccolella. Simone is one of the organizers of the workshop and is currently a postdoc in our university. He is an expert of combinatorial methods for pangenomics, and so we are sure that we are going to conclude the workshop on a very high note. So please, Simone, start your presentation.
Thank you very much, Gianluca. Thank you all for attending this workshop. This is the last one. We are close to lunchtime, so I'll be quick. And my talk would be on modeling and combinatorial approaches for pangenomics. Okay? What does it mean? I will continue on the same note that Davide left us on, which is more a computer science approach to this biological problem. And I would like to show you 3 different methods that we developed in our lab, with a focus mostly on how do we model biological problem in a more computer science way, and how can we solve them algorithmically. So I will not go into deep learning or machine learning topics. This would be just purely algorithmic and combinatorial. However, my goal is not to be too technical. I don't want to bore you with the technical details. I just want to share with you some idea and key concepts that we can use, hopefully. So pangenomic construction is the first problem I would like to show you. I will not go into the details of what a pangenome is. We already seen it described multiple times in this workshop. But what I'm referring to is, just to be clear, it's a graph pangenome, okay? And typically there are different ways to do so. One of the first ways that we use to build pangenomes, and some tools still use this method, is to use a reference plus a VCF file. So basically, we would have, in this case, a reference represented by this line, and then we have different variations, okay? And for example, in this case, this red one would be considered as deletion, okay? And what we end up is that we want to encode these genomic variants represented in our VCF file as different nodes in the graph. So we try to keep our graph as linear as possible, but we had different nodes and different links when it is necessary. This is one of the easiest methods to do such a construction, because it relies on data that we pretty much already have. We already have reference, we have multiple VCF for different populations. It's good, but it has some issue. Why? Well, because the main goal of the pangenome is to try to avoid what we call the reference bias. And if we rely on the reference and a VCF to construct a pangenome, in some sense, we are still relying a lot on the reference, and we still may have this so-called reference bias. Therefore, different methods have been proposed. One, which is the one that we will focus the most, is the one based on the multiple sequence alignment. So what we do is that we take a bunch of sequence and we do a multiple sequence alignment. So we have different methods to do so. We typically either do a pairwise mapping, and then we map all the pairwise all together and obtain a, we can call this as a matrix, right? We have this matrix where we have different organisms as rows, and we have the different genomic composition on the columns. This is technically better than what we have before, because at least we try to minimize reference bias, since we don't have a reference right now, but we are comparing all the organisms between each other. The main drawback of this is that multiple sequence alignment is a costly operation. It requires a lot of time and a lot of resources, especially as the length of the genome increases and as the number of sequences that we want to align increases. And similarly to what we did before, we are able to construct a pangenome from this multiple sequence alignment in many different ways. Every tool has its own way to implement this, but the idea again here is to keep the common parts all together and then make adjustment when needed. There are other ways to do pangenome construction other than doing proper multiple sequence alignment. We can do pairwise mapping and augment at each comparison the graph. We can also do some incremental construction, which is not dissimilar to what we've seen in the talk yesterday, where we have a new organism that we want to, a new individual that we want to add to our current pangenome, and then we adjust the pangenome based on what we see. This is one of the tools that we proposed. It's called PangeBlocks. This has been published last year in BMC Bioinformatics. And here we relies on two different concepts. So we are going to use multiple sequence alignment, and we are going to use an ILP to solve this problem. Thanks to Davide, to his previous talk, we now know what are these maximal blocks. And in fact, we used a similar approach in this work where given a multiple sequence alignment, so we're not as before with a matrix of 0 or 1. Here we have different characters, and we want to compute these maximal blocks. So as explained by Davide, we want to have blocks that cover the more position as possible in both columns and rows. And the idea is that we want to cover the entire multiple sequence alignment with these maximal blocks. Now, technically, maximal blocks cannot cover all of our input. Therefore, we might have to not really use maximal blocks, but use smaller blocks to cover more. And here there are trade-offs that we can have, and we'll see them shortly. So what is the ILP modeling? So ILP, as you all know, is integer linear programming. So what we want to do is that we want to devise a mathematical formulation to solve this problem. And in particular, with any ILP model, we were going to have some decision variable and some constraints. So the basic of this is that the decision variable are binary variables in this case, where we want to see, we have variable for each block, okay? And the decision here is whether we want to use this block or not. And we have some constraints, which is, for example, each cell must be covered by only one block. Therefore, with this, we can construct a model such that each of this cell of the matrix will be covered by one block. Of course, this alone is not enough, right? Because the easiest solution would be to have a block for each of these cells, which is not good. Therefore, we also have some objective function. And I think this is the beauty of this work, alongside the actual ILP formulation, is that basically once we have this variable, we can define whichever cost function that we want. So if we have a look at this slide, C would be W and C would be the cost, sorry, not W, omega, and C would be the cost of this function. And this can be defined as we want, right? So for example, in our paper, we have a bunch of those. I'm going to show you 4, which is the number of nodes. So for example, in this case, we want to minimize how many nodes we have in a graph. Or we can minimize the length, the total length of the graph. We can also prioritize, in some cases, shorter nodes, or either way, we can minimize nodes that are too short. Or we can minimize nodes that use few sequences. Okay, so this is 4 of the objective function costs that we define in this paper. And while they tend to have some similarity between them, all of them can be used to optimize a different part of the graph such that we will result in different topology. Slightly different topology that still explain the same input. The beauty of this is that, well, first of all, it's easy to add new objective function. So if we have some specific cases for which we would like to have a specific function to optimize, we can do so just by changing our objective function cost, while the other entire modeling still continue to work. This is quite nice, and it is nice that this can be fine-tuned for each specific case that we want to study. This is nice, but still it is an ILP. So even though I really like ILPs, I think they are a beautiful modeling approach, they have a main drawback, which is computational cost is very expensive, both in terms of memory usage and time, okay? This is not always the case, but in average, they tend to be quite expensive. Therefore, this is still a work in progress. We have made some approaches, but none of them, we haven't found the correct method yet. But the idea would be to devise an heuristic method to solve the same problem. And this, of course, would mean that we lose the guarantee of optimality that we retain with an ILP, but with the main advantages of gaining a lot of performance to solve the same problem, right? Okay, so suppose now that we solved the pangenome construction and we want to use it. The first approach that comes to mind, the first idea that comes to mind when we want to use a pangenome is to do, for example, a sequence alignment. Typically, we all know how to do sequence-to-sequence alignment. That's what we usually do when we want to analyze and to map reads. In this case, the problem is a bit more difficult, right? Because we have to do a sequence-to-graph alignment. So suppose we have this string and input. We will go step by step, and you will see that it's quite easy, right? Because we can start with T, we can take T, then we can take ACG, which is the next node, and we can continue this mapping as long as the sequence we want to map can be found in the graph. Of course, if we have insertion, deletion of error, we can modify something. What is something? Well, usually in most of the cases, what we actually modify is not the graph, right? But we want to modify the read. So we would obtain some different cigar, which is what we usually do when we do sequence-to-sequence alignment, right? So in this case, we do not get exact coordinate position as we normally do, but we will get this sequence of nodes in the graph that would be the mapping of our read. Now, what happens when we have a variation graph? Well, what is a variation graph? It's something that we add on top of this structure. So, for example, here I've added some colors to this graph, and this graph represents now what we call a variation graph, where we basically have different walks or paths in this graph, and they represent something. Okay, typically they represent haplotypes, but they can represent other type of information that we want to encode in the graph. So in this case, this is what we would have. So we have for each of the, let's say in this case, they are haplotype for each of these walks in the graph. We have different color and different nodes. So what happens when we want to now sequence, so to map a sequence to this graph? Well, if we have a a sequence like the one we had before that perfectly matches any of the haplotype, we have no issue at all. This is all on the green haplotype and we're good to go. What happens when we have something that does not map correctly? Now we have different way to solve this problem, right? What we can do is that we can follow the paths, okay? And in that case, we will prioritize the paths of the graph. So for example, we want to map following one of the haplotypes. Here we have different ways of doing so. For example, here we have a deletion or a substitution or an insertion, depending on which path we choose. And of course, we would have a scoring function for these, and we would choose the best one for this. This is nice because it makes the problem easier. It's not that different from doing sequence-to-sequence alignment. We just have to do it multiple times following all the paths in the graph. However, we are not really fully utilizing the capability of a variation graph. Another way we can do it is that we ignore the paths, right? We do not consider them, and whatever comes out based on how close the sequence are to the graph. So we do like we did before, and we ignore the paths. That is still easy problem, but now this will require some post-processing because we have to decide what we do with these paths. We can have multiple paths, and then we have to decide which one we want. Or another approach is that we, on top of the original sequence to graph alignment, we can model recombination. So in this case, we would call a recombination moving from one path to another one in the graph. And that's what we did in this paper. RecGraph is a recombinational aware alignment of sequence to variation graphs. So the goal of this is that we want to map following the paths as much as we can, but we model moving from one path to another, and we have a cost associated with that, depending on the goal of what you want to have, right? So we can customize the weights of these operations. Very interesting in this paper, and if my memory serves correct, there is no other paper, no other method that allows to do this, which is this sort of displacement where we can move from one path to another at any point in the graph. There are methods that allow to move to one path to another, but only in nodes where the two paths collide. But with this method inside a bubble, for example, we can move from one path to another. Of course, this adds a cost to our sequence, also cost that we can fine-tune to our applications. I will not go into the detail of this, but if you want, the paper is out to read. This is dynamic programming approach, and we want to maximize a given scoring function. All these ways can be fine-tuned if you want to minimize substitution, insertion, deletion, recombination, or different displacement values. And in this work, we also tried this on bacteria. And in particular, what we try to do with bacteria is that we try to assess also with phylogenetic analysis whether this sort of recombination made sense or not. And in most of the cases, they do, and we actually find recombination to different organisms. These are simulated results in this case, but we simulated them with a specific recombination in a specific point, and we were able to actually reconstruct the same recombination point, which is good. Now, this work has a limitation. Currently, it's limited to only one recombination, and this is done optimally. And again, optimality requires a lot of resources. So what we are doing right now is to develop an A*-based heuristic to do this mapping. And also we want to allow multiple recombination such that it would be less computationally intensive. This work is not published yet, but we have a preprint that you can have a look at. So the last problem that I want to show you is the problem of alternative splicing. So alternative splicing is that this phenomenon that we see in nature where when we basically express from DNA, RNA, and then we get the protein, this translation is not always the same, right? We have, given the same DNA, we can have the, based on the condition of the replication or the environment, the same portion of genetic material can produce different mRNA and therefore different protein. And the way we usually solve this in classical approaches is that we have alongside our reference a database of all these different exons and how they combine with themselves to produce proteins. However, this is still reference-based, and all the databases are reference-based. But let's have a look at what we see usually. So suppose we have a reference, we have it annotated with this part of the reference are the different exons, okay? What we do is that we get a bunch of reads. These are spliced reads, spliced aligned reads. So we have a bunch of them. What we do is that we try to separate them in cluster and see what they actually express. Okay, so in this case, the first one would be 1, 2, 4, the second 1, 3, 4, and the last one would be 1, 2, 3, and 4. This is all well and good. And we can classify these different events based on what happens. So for example, if in the annotation we have this pattern, so we have exon 1, exon 2, and exon 3, we see the reads that skip over exon 2. We call this an exon skipping. Sorry. If we have the two exons, but we also see a retained part in between those, so we would call this an intron retention. So where we basically, our RNA also express the part in between those exons. We can also have points in which, for example, one exon extends over what is in the annotation. And in this case, we would call this an alternative splice site, in this case on the 5 prime. And similar, we can have the symmetric on the other side and have an alternative 3 prime splice. So how do we do this on a pangenome? This is quite new. This paper is, I think it's out. If not, it will be quite soon on PLOS computational biology. And as far as we know, this is the first method to actually do a proper modeling of this event on a pangenome. So what we do is that we want to augment the pangenome with this type of information. So let's start with the reference, just to give you an idea. So we have this reference, we have the variation in the reference, and then we have different annotation for the transcripts. First of all, we also get a pangenome. We also need a pangenome. What we want to do is that we want to up the information regarding the transcripts on the pangenome itself. And therefore, we have developed this technique that basically try to transpose the linear information on a pangenome, and we can add on top of the already present information, we can have additional information regarding these splicing events. We can also annotate this splice pangenome by including into it the actual information on the transcript and the haplotypes so that we have all the different possible combination inside of our genome. Therefore, once we get a read and we align this read with a spliced alignment, we can follow what happens in the graph and actually deduce the type of event that we see. So the main modeling contribution of this work is that we define a framework when we can basically call these different events. So if we start with annotated events, this is a different representation of the exoskipping that we saw before, but is actually saying the same thing, but instead of a linear reference, this is done on a pangenome. Unfortunately, it seems like I cannot use a pointer, but basically we have the blue squares are the exons, the small white squares, unlabeled ones, are the introns, and then we have these edges that represent the different splicing events on top of the edges that represent the actual genomic pangenome. So we have exoskipping, we have the 5' splicing site that you see is expanding on the right. Similarly, the 3' is expanding on the left. And intro-retention, here we see that we have a bigger transcript that comprises all of the nodes. This is for annotated events, and we basically get this information from a database, but it also works for novel events, so events that we don't really have annotated and we discover when aligning the reads. I will not go into the details of this modeling because it's too complicated to see this in a slide, especially without a pointer. We tried this method, we tested on both simulated and real data, and we compared it against reference-based methods that are the only methods that we have. We don't really have any other method to do so on the pangenome. And we actually showed that using a pangenome is first of all feasible, and we obtain very good accuracy, both in precision, recall, and all the measures that we tried. So not only is possible to do it on a pangenome, but we can do it with high confidentiality of our results, and especially when we compare with the with the other methods, also on true validated events in the lab. This is all good. The main drawback for now is that these methods rely on having a splice alignment to a sequence graph or to a pangenome graph, let's say. And currently, there is only one method that allows to do so, which is VG-MPMAP. And that method requires a lot of resources. So, but our tool is independent of the type, is agnostic on the type of splice reads that we obtain. So we hope that when more and more people start working on this type of alignment, better and faster alignment tool will come up, and we will still be able to get even more out of this work and even more out of the pangenome. So now this is the most important part of the talk. So the boring stuff is over. Advertisement time. What are we going to do right now? So these are the next events that we have. The first one is CPM 2025. This is going to be in Milan, in Italy, and June 17 to 19 this year. You're all welcome to participate, and I think the submission deadline is past, not sure. But if you got accepted or if you want to join us in this beautiful city, you can come and we would be happy to host you. Now, more specific for the 4Genome project, which is the sponsor of this workshop, we will have a PhD school plus an hackathon this year in July. This date should be correct, meaning that there is the time frame. It's not strictly defined yet, but if it's not this exact date, it will be around that date. So would be not that far to that. So we will have 4 days PhD school, and the topic would be Introduction to Bioinformatics Services in Pangenome, Cancer Genomics, Introduction to Modern Genome Sequencing Technology, Data Analysis and Software Tools in Genomic Structural Variation, the latest trends, methods, and experimental validation of this data. And we will have a very nice, I think, part that would be especially useful for early postdocs and PhD students, whereas in a more generic way, not specifically for research, but what are the research challenges, how to do this is really hard, entrepreneurship. And this is going to be trained by Amble, which is one of the partner of this project. And as I'm pretty sure you all know, they're quite good and quite strong in what they do. So it would be very interesting. And following this, we will have a 3-day hackathons. This will be on genomics, proteomics, and bioinformatics. For now, we have some confirmed speakers for this school, which are Piotr Prince, Eric Garrison, and Enza Colonna, all of 3 from University of Tennessee. And you may know them. You most likely know at least Eric Garrison, which is one of the main proponents of the VG toolkit for variation graph and for pangenome. And we're going to have Tobias Rausch, which is a bioinformatician from Amble. Lastly, thank you for attending this workshop. The live streaming recordings are already available on the website. You have the link. We sent to you in both in email when you registered. It's on the website, and we sent on the metrics. And for now, these are just the live streaming recordings. Edited video will be available soon. We hope next week. And lastly, thank you all for attending to this talk and for attending to the whole workshop in general. I would like to thank 4Genome2, which is the sponsor of this workshop. And these are the partners of the project, which is Comenius University, Amble University, the University of Milano Bicocca. And thank you for the European Union to funding this project. So if you have any question, I'm happy to take them.
Thanks, Simone. It was very clear presentation of the current status. One quick question is, what of the several problems that you have described, which one do you think will be the most challenging in the next few years?
I think both the most challenging and the most important would be the sequence to graph alignment. The reason why I say this is that we have a lot of methods that build pangenome, and I mean, pretty much they all give us some solution which is good in practice. But as we've seen with reference genomes, we know that the main goal, the main challenge is actually to get an alignment of this sequence, because this is what all the downstream analysis rely on. And we saw a lot of methods to do sequence to graph alignment, but none of them still solves all the issue. We don't really have a clear understanding on what we want exactly from the graph and what we want this alignment to be. And this is why we see a lot of alignment tools. And each of them has its pros and cons. it is specifically devised for a particular problem, and either we want to have more of those to solve specific problems, or we may have an established one that can encompass all of these issues. But right now we don't really have a de facto standard, and I think that would be the most important and the most challenging, because it's hard to do so.
Okay, thank you very much. It was very clear, very relevant. I think that we can conclude this first annual workshop. Thanks everyone for participating, and I'm sure you will disseminate these videos once they are fully published on YouTube.